Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Understanding alpha-actinin, structure, isoforms, and mechanisms. Table of Contents. Introduction. Actin parent molecule. Alpha-actinin functions. Alpha-actinin types. Alpha-actinin structure. Regulation by different mechanisms. Conclusion. Introduction. Alpha-actinin is an actin-binding protein that interacts with filamentous F-actin but does not bind to globular G-actin. It is ubiquitously found in the cytoskeleton of both muscle and non-muscle cells, where it plays a crucial role in organizing and binding actin filaments. In muscle cells, it is calcium insensitive. In non-muscle cells, such as neurons and fibroblasts, it is calcium sensitive. As a structural component of the cytoskeleton, alpha-actinin belongs to the spectrin superfamily and is distributed in various cellular regions depending on the tissue type. Its key functions include stabilizing the contractile apparatus in muscle cells, modulating receptor activities, acting as a scaffold to connect cytoskeletal proteins involved in cell signaling pathways. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. The presence of alpha-actinin at multiple subcellular locations, such as cell protrusions, cell matrix contact sites, and stress fiber-dense regions, highlights its role in linking cytoskeleton components with other cellular proteins. It exists as multiple isoforms, which have been genetically and biochemically conserved across mammals, protists, invertebrates, and birds. Mammalian cells exhibit the greatest diversity, with 4 alpha-actinin encoding genes, giving rise to at least 6 distinct protein isoforms, each with specific tissue distribution and expression patterns. Actin parent molecule. Actin is the most abundant protein in eukaryotic cells and is highly conserved throughout evolution. It engages in more protein-protein interactions than most other studied proteins. A key component of the muscle contraction apparatus, actin filaments consist of repetitive actin protein units in 2 forms: G-actin, globular, monomeric form; F-actin, filamentous, polymeric form. As the fundamental building block of the cytoskeleton, actin facilitates various cellular movements, including maintaining cell shape, enabling cell motility, regulating transcription. Actin filaments interact with myosin during muscle contraction, with tropomyosin proteins regulating their fusion in the process. Through cellular and subcellular movements, actin dynamically regulates the structure of different cell types. Alpha-actinin functions. The primary function of alpha-actinin is to cross-link actin filaments and other cytoskeletal components, influencing cellular movement, cohesion, and mechanics. It provides structural stability to actin filaments and serves as a scaffold, integrating the cytoskeleton into signaling pathways. Alpha-actinin interacts with approximately 30 cellular components, with its isoforms playing an active role in nuclear events. It is highly active in polarized cells, where extensive actin polymerization facilitates cell migration and adhesion. As the main crosslinker in stress fibers, it is abundantly present in dendritic spines, contributing to focal adhesion and neuritic growth. Its interactions determine the localization of alpha-actinin to cellular compartments, including plasma membranes, transmembrane receptors, and membrane lipids. Alpha-actinin types. 4 types of alpha-actinin regulate cytoskeletal activity in muscle and non-muscle cells. ACTN1, encoded by the ACTN1 gene. In skeletal muscle cells, it facilitates Z-line attachment to actin myofibrils. In smooth muscle cells, it connects dense bodies to actin filaments. ACTN2, encoded by the ACTN2 gene in humans, composed of 894 amino acids with a molecular weight of approximately 103.8 kilodalton, expressed in skeletal and smooth muscle cells, anchors filamentous, actin fragments to Z discs through phospholipid mediation. ACTN3, encoded by the ACTN3 gene, also known as the sprinter gene, located on chromosome 11, expressed exclusively in type 2 muscle fibers, plays a role in enhanced athletic performance and muscle injury recovery, particularly in elite athletes. ACTN4, encoded by the ACTN4 gene, involved in cytoskeletal regulation related to metastasis. Mutations in ACTN4 are linked to kidney glomerulosclerosis. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Alpha-actinin structure. Alpha-actinin is an anti-parallel homodimer that plays a crucial role in cross-linking actin filaments within cells. It belongs to the spectrin superfamily, characterized by 3 main structural domains. 1. N-terminal actin-binding domain, ABD. Second rod domain, spectrin repeats. Third, C-terminal calmodulin-like, KAM domain. First, N-terminal actin-binding domain. The N-terminal actin-binding domain is the most conserved domain among spectrin family proteins and is essential for binding actin. It consists of 2 tandem calponin homology domains, calponin homology 1 and calponin homology 2, each composed of approximately 100 amino acids arranged in 4 alpha helices, A, C, E, and G. Helices A and E are located externally, while helices C and G are positioned internally. Unlike other spectrin proteins, alpha-actinin N-terminal actin binding domain contains only 2 minor helices, B and F. The calponin homology 1 and calponin homology 2 domains interact through a combination of polar and hydrophobic forces, stabilizing the interface. Calponin homology 1 alone has a lower affinity for actin binding, while calponin homology 2 is insufficient for cross-linking actin filaments. Together, they enable strong actin interactions. Second rod domain, spectrin repeats. The rod domain, composed of spectrin repeats, determines the protein's length, flexibility, and ability to cross-link actin filaments. In vertebrates, the rod domain typically consists of 4 consecutive spectrin repeats. The number of repeats varies across species. Protozoans have 1, fungi have 2, and parasites have 5. These repeats originated from a single precursor protein through 2 intragenic duplication events. Each spectrin repeat adopts a 3-dimensional helix-coiled bundle structure with short, rigid helical linkers, providing structural stability necessary for actin-bundling. The overall length of the alpha-actinin rod domain is approximately 240 angstrom, with a width of 40 to 50 angstrom. Spectrin repeats are primarily composed of hydrophobic amino acids, such as leucine, isoleucine, and valine, which contribute to protein stability. Proline and glycine residues are also abundant, ensuring flexibility and maintaining the protein's shape. Third, C-terminal calmodulin-like domain. The C-terminal calmodulin-like domain is involved in calcium-dependent intracellular signaling and is located at the protein's C-terminus. It consists of 2 pairs of EF hand motifs, EF1/2 and EF3/4, which are helix-loop-helix structures capable of binding calcium. Upon calcium binding, the globular domain shifts from a closed to an open conformation, exposing hydrophobic residues that enable interactions with specific targets. Alpha-actinin isoforms differ in their calcium sensitivity. Muscle isoforms lack calcium binding ability due to evolutionary changes in their EF hand motifs. Instead, they are regulated by phosphatidylinositol 4, 5-bisphosphate. Non-muscle isoforms, found in fibroblasts and neurons, retain calcium binding ability, which regulates actin-binding activity in response to intracellular calcium fluctuations. During evolution, the ancestral alpha-actinin gene initially encoded a calcium-sensitive isoform. However, muscle-specific isoforms later emerged, losing their calcium sensitivity to allow actin-binding regulation independent of calcium signaling. Regulation by different mechanisms. Alpha-actinin activity is regulated through multiple mechanisms, including post-translational modifications, protein-protein interactions, and intracellular signaling pathways. These regulatory mechanisms modulate alpha-actinin's interaction with actin filaments and influence cellular processes such as cytoskeletal organization, adhesion, and motility. Two key regulatory mechanisms, 1. Phosphorylation, and 2. Proteolytic cleavage. First, phosphorylation. Phosphorylation involves the addition of a phosphate group to a protein, altering its activity and interactions. In the case of alpha-actinin, phosphorylation at specific sites can either enhance or inhibit its association with actin filaments. Activation. Phosphorylation at certain residues promotes the binding of alpha-actinin to actin filaments, stabilizing the cytoskeletal network. Inhibition. Phosphorylation at other sites reduces actin-binding affinity, leading to cytoskeletal remodeling. The effects of phosphorylation depend on the specific kinase enzymes involved and the phosphorylation sites targeted. A notable example of phosphorylation-regulated alpha-actinin function is observed in focal segmental glomerulosclerosis, an autosomal dominant disease caused by a mutation in Alpha-Actinin 4. Studies indicate that phosphorylation of tyrosine 4 and tyrosine 31 in Alpha-Actinin 4, mediated by growth factors in a hierarchical manner, significantly reduces its actin-binding ability. This regulation helps prevent excessive cytoskeletal aggregation while promoting cell motility. 2. Proteolytic cleavage. Proteolytic cleavage is another crucial regulatory mechanism that affects alpha-actinin function. This process generates smaller protein fragments with distinct activities compared to the intact protein, influencing actin filament organization and stability. N-terminal cleavage. The N-terminal cleavage of alpha-actinin 4 at tyrosine 13 and glycine 14 does not impair its actin-binding function. Instead, it enhances actin bundle arrangement, facilitating cell migration. C-terminal cleavage. Cleavage at the C-terminal of alpha-actinin 4 or other alpha-actinins reduces calcium-dependent actin binding, thereby impairing focal adhesion and cell motility. One of the key proteases involved in this process is calpain, which plays a role in T-cell activation. Upon CD3 and T-cell receptor stimulation, calpain-mediated cleavage of alpha-actinin enhances cytoskeletal plasticity, facilitating immune cell spreading and migration. Conclusion. Alpha-actinin serves as a multifunctional, actin-binding and cross-linking protein, playing a crucial role in cytoskeletal organization. It facilitates the bundling of actin filaments, stabilizes the cytoskeletal network, and contributes to cellular functions such as adhesion, migration, and signal transduction. Additionally, alpha-actinin is involved in membrane translocation and intracellular protein trafficking within signaling pathways, highlighting its significance in cellular dynamics and structural integrity. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!